30-year-old male patient presented with a history of UTI, the cause of which is suspected to be bacterial. Which of the following is used by the bacteria for adherence? So that means they are asking what is the organ of adhesion? What is the organ of adhesion in bacteria? So to understand this, let us look into the morphology of a bacterial cell. So let us come from inwards to outwards. So this particular thing which is called is the genetic material of the bacteria, and the genetic material of bacteria is supercoiled double-stranded DNA material. So supercoiled double-stranded DNA material, which is circular, is the genetic material of a bacteria. So next, all these are inclusion bodies, which will be the storage bodies or the storage granules of the bacteria. So these are the ribosomes, the sites for protein synthesis, and this is the cell membrane, also called as the plasma membrane. So this is the plasma membrane of the bacteria, where at points this plasma membrane can also show invaginations like this. So they can show invaginations from the plasma membrane, and these invaginations of the plasma membranes are called as mesosomes. So mesosomes are the invagination of the plasma membrane, where they are involved in respiration of the bacterial cell, and they are more in quantity in case of gram-positive organisms. Okay, so all these are the potential questions where they are more in number in gram-positive organisms. So next is this particular one. This is the cell wall of the bacteria, which we will study in detail. This cell wall is covered by a thick material, where this is called as the capsule, and these are the cell appendages. One is the flagella, the longer one. The other one is a shorter one, which is fimbriae, that is organ of adhesion. We will look into all this in detail. So we have finished with genetic material, plasma membrane, mesosome. Now we will study about the cell wall of the bacteria. So the cell wall of the bacteria should be studied in terms of one as gram-positive organism, and the other cell wall is that of gram-negative organism. Okay. Gram-positive organism, I'll write like this. So positive organism, P is something which is very important for us. So in gram-positive organism, we have very thick layer of what is called as peptidoglycan layer. So peptidoglycan layer is very thick. Just look into the gram-negative cell wall, 1 to 2 layers thick. But here say 10 to 15 layers thick peptidoglycan layer. So the very first point of importance is peptidoglycan layer, okay, which is very thick. And the second one is, if we study the internal structure of this peptidoglycan layer, we have bridge like this, and these bridges are the one which makes them, give the property to them for gram positivity along with peptidoglycan, and these bridges are called as pentaglycine bridges. So the second point is pentaglycine bridge. Okay, so the two things which are important in gram-positive organism cell wall is with their peptidoglycan layer, which is very thick, and the pentaglycine bridge in peptidoglycan layer, where both of this contributes to gram-positivity. Okay, so next let us come to gram-negative cell wall. So I want to observe this particular part of gram-negative cell wall, which is not there in gram-positive cell. Right? Okay. So we will study about the same in detail by this image. So first point is, as we saw, the peptidoglycan layer, which is very thin. The second point is the whole chunk from after peptidoglycan layer, which is positive only in gram-negative organism and not there in gram-positive organism. What are these layers? The first layer, which is not present in gram-positive, is the outer membrane. Outer membrane followed by the second one, which is lipopolysaccharide layer. So this lipopolysaccharide in turn is made up of lipid A, core polysaccharide, and O side chain. Okay, so we have outer membrane, we have lipopolysaccharide layer, where the lipopolysaccharide layer is in turn made up of lipid A, core polysaccharide, and O side chain. So what is the potential question here? First question is, outer membrane is present in only gram-negative organism. The second thing is, from which is O antigen derived from? So O antigen is derived from lipid A portion. So kindly note, O antigen is derived from lipid A portion of the lipopolysaccharide layer. Okay.
To watch the full video, download our app on App Store and Play Store.